And you want to introduce Carla? You might as well. You know her so
Sure,
well.
yeah,
Okay.
but I'll still probably read it.
Okay, cool. All right.
Okay, we're lucky today to have Carla D'Antonio on the line. She wanted to be here in person, but unfortunately, Carla, what happened to you?
I broke my
Are you there?
ankle and shin.
She broke her ankle and shin. So she's in bed with probably her leg elevated, and she's so hardcore that she's here with us today. Well, sort of in spirit, like coming through. Okay, so for those of you who don't know Carla, she's the professor and chair of the Environmental Studies Department at UC Santa Barbara. She works on many different aspects of understanding plant change, and she also enjoys using ecological knowledge to address management and restoration questions in the ecosystems of California and Hawaii. All right, Carla, take it away.
Okay, thank you. And I'm really excited to be here. And I hope this is the last time I give a talk from a horizontal position. Well, before I begin, can everybody hear me? Just shout out yes or no. Is it good? Yes. Okay.
It's a
Go ahead, go ahead.
Okay. So before I begin, I want to thank my collaborators, mostly my students and people in my lab, for their feedback and input and for helping me gather some of the traits that I'll talk about later. And the IPIF here stands for Institute of Pacific Island Forestry, where I work in Hawaii, and the contribution of some people there will become clear later on. So Hugh yesterday gave you a brief primer on what restoration is, and I would say that the primer he gave you is more relevant to what Eric Higgs called Restoration 1.0, which is what we did for maybe 25 years, where we used history as a template. We built communities that were based on what we thought the historical community was, and we set them on a singular trajectory based on composition. But in the last 10 years, with the embracing of the fact that climate change is so real to us and that we really can't go back, we have what we call now Restoration 2.0, where we just use history as a guide. We set ecosystems potentially on multiple trajectories, and we focus on process rather than just on composition. And we base that process and the composition on pragmatic goals that make the restoration possible. So with that as a background, let's talk about what we know about chaparral restoration. And I want to make reference to a chapter that I was a co-author on with Edie Allen and many others, in which we reviewed what we know about chaparral restoration. And the conclusion we came to is that it's extremely challenging, that seeding of chaparral species in virtually all cases fails, and outplanting and irrigation are really necessary, which limits the size and the logistical issues you can overcome in restoration projects. And what we also concluded in this review was that the reality is that chaparral restoration in most cases really results in sage scrub restoration. That is, we're putting sage scrub species back instead of chaparral species. And keep that in mind as we go through this talk, because maybe that is what we can do. I just want to make reference to a poster by Stephanie Ma about that we have experienced many of the same challenges that we came across in reading the articles in that referenced in that chapter. We've been doing a restoration project in Peru on the Los Padres. Seeding has been completely unsuccessful despite tens of thousands of seeds thrown out on the landscape. And we face severe grass competition from the woody seedlings we put out face severe competition from grasses. So the two questions I want to address in this talk today are, briefly, what are the environmental conditions where chaparral is being lost? And then second, how might we use species traits to define restoration combinations or trajectories in these habitats? Now, this first question, although it's been addressed somewhat by Keely and Safard in the talk earlier today, is important because it defines the conditions where most of the restoration will occur. So to get at this question, we took an approach that of taking the VTM maps, that's the 1934 maps of Ventura and Los Angeles counties, and mapping what's shown in gray here, all of the chaparral in those maps. Then going to the 2010 maps and looking at where chaparral remains, shown in green, where chaparral turned into something else, and we have two something else's that it turned into, either sage scrub shown in purple, or grasses or herbaceous vegetation shown in orange. And some of this overlaps with short interval fire periods. So we then used, and this is work that's being done by Shane DeWeese, and he has a poster which I encourage you to go look at. We evaluated, using a random forest approach, the predictors of conversion either to sage scrub or to grassland. And the things we considered were precipitation, elevation, distance to coast, maximum August temperature, minimum January temperature, distance to roads, some fire things, and several other environmental characteristics. So here's what a graph looks like from a random forest analysis. These are importance values of the factors. And you see that for predicting the loss of chaparral to sage scrub, elevation is the most important predictor, followed by precipitation. And both the summer temperatures and the January temperatures are hotter in the sites that converted. So this conversion is happening at low elevation in dry, hot areas, much like the results that Safard and Keeley showed from the Santa Monica Mountains. If we look at predictors of conversion to herbaceous vegetation, again, we see a similar picture. Lower elevation sites more likely to convert, drier sites more likely to convert, closer to the coast, and warmer winter temperatures. And if you want the actual mean values of converted and unconverted sites, go see Shane's poster. So basically what this tells us, I just want to make a quick comment about fire return interval. It is significantly different between converted and unconverted sites, but it didn't show up as important a predictor as these basic habitat variables. So what this says to us is the conditions where most restoration is going to be going on, where chaparral is being lost, are hot, dry, low elevation slopes. They were closer to roads, significantly closer to roads, and this means they have a higher probability of invasion by exotic species. And it also means that the native species pool, when you look at these sites and try to restore them, is going to be biased towards sage scrub species to begin with because of the generally lower distribution of sage scrub. So here's an example of one of these sites. This is one of our sites that we're working on restoration in Piru, above Lake Piru. And it's exactly one of these areas. It's hot, it's got very low mean annual precipitation, it's close to a road as shown here, it's got lots of exotic species, particularly Avena and Bromus diandrus. So what approach can we take given that we know that seeding is unsuccessful? We can't, these sites are very difficult to access. We can't set up irrigation systems there. And we decided to try a plant functional traits approach. And by functional traits, I mean traits that influence and correlate with the growth, reproduction, and survival of a species. For example, you could imagine on the x-axis of these graphs, you see wood density. That would be a plant functional trait. And on the y-axis are some aspects of demography, so growth rate or mortality rate. And these are for a lot of different species from some work by Peter Reich at Minnesota. And you see this correlation between that functional trait and some important demographic parameter. So functional traits vary among species and environments in predictable ways. So we decided to try to use them to build communities based on traits. So they're essentially shortcuts for ecosystem functioning, and maybe they allow us to take a more quantitative approach to selecting species for restoration. And just as an example, if your goal in restoration is fire tolerance, you might look at and measure a whole bunch of traits related to bark thickness, water content, resprouting ability, and other traits that relate to fire tolerance. or likewise with drought tolerance, some leaf size traits, water use efficiency traits, and so on. So the tool that we're using to do this is just exactly what a tool should be, a simplification of multivariate analyses. And it's called REST, or Restoring Ecosystem Services Tools. And this is where the Institute of Pacific Islands Forestry comes in. Susan Cordell, who's the lead scientist there, and a plant ecophysiologist, has developed this tool, and with Becky Ostertag, done a lot of work looking at what traits would relate to what particular restoration goals. You can find access and download it from a website where they've been working on creating hybrid ecosystems. So quickly take a shot of that if you want it. Okay. So how do you run this? What do you need to do? First, you have to articulate your goals or your objectives and your constraints. Like, are you shooting for drought tolerance? Are you shooting for maximum diversity? What is it that you want? Then you have to select the appropriate functional traits, determine the pool of species for trait sampling, collect the trait data. So these two things are in red because these are really challenging aspects of this approach. Then you run a principal components analysis and evaluate all your species that you've put into your database in this trait space. Then you filter the principal components analysis, and you consider propagation issues and other constraints once you start to have your PCAs. So let's run that through for this project. Our goal in Piru is a diverse native perennial cover that supports healthy food webs, reduces erosion, and is resilient to drought and fire. That's pretty pie in the sky, but it seems like a good set of goals to me. So our objectives are to establish shrub assemblages that would meet these goals, that will suppress exotic herbaceous species, and will be resilient. Now, our constraints are that we can only collect plant material that's in the region, and all the species must be native. There could be some great exotics that will do well out there, but we don't want them. Irrigation is not possible, access is challenging, and rainfall is unpredictable. Okay, what traits did we decide to include in this analysis? So we pick out traits that are relevant to particular goals and constraints, things like leaf thickness, leaf mass, leaf N, C, etc., because these relate particularly to drought, which we're interested in. Now, we have to also select by traits that are easily measured. There are plenty of curves for P50 curves, like Steve Davis made reference to yesterday, out there, but there's a really small subset of species for which those curves exist. So when it gets right down to it, you can't use really detailed ecophysiological data like maximum photosynthesis or P50, because you'd have to have that on 25 species, and that's not practical at this time point. So we focused on more easily measured traits, and we tried to pull traits also from the literature or from various databases. The REST program includes 116 traits, and you can add more to the program if you want. And some of the ones we focused on are shown in bold. As I said before, there's built-in vegetation goals, and if you click on one of these goals, such as drought tolerance, a group of traits pops up. For example, restoration goal drought tolerance, shown up here. You get a whole list of traits, and you click on the ones that you have out of the ones that are in their database. You could add more to it, so you can add new traits, see down here. You import your whole traits matrix from an Excel file as a CSV file. You cannot include a trait if you don't have a value for every single species in there. Here's another restoration goal, fire tolerance. You get some of the same, but a lot of different traits as well. So how do we determine the pool of species? Well, we went out to the sites and we hiked and drove all over and looked and thought, okay, what might the pre-degradation composition have been? And we found a few places that maybe represent that, and post-degradation composition, and natives from elsewhere that are nearby. Just to give you a visual feeling, this is what the pre-degradation native woody species community seems to look like. And here's what the native woody residents today look like. And so right away you see a huge difference in what these species look like, and that's going to be reflected in the traits as well. So here's a list of our species, and I put the ones in red that are more of what you consider to be soft leaves or sage scrub species, and the other ones are more sclerophilous chaparral type species. So we had a total of 23, I think, species for which we were able to get all the traits. So we had 34 traits that we collected, and then this is where it gets fun, we run the PCA. So here's a principal components analysis with all the 23 woody species and all the traits, just to see what happens if we just use all the traits together. Where do our species fall out? You don't know what these numbers are here, but I do. And I'll tell you, this clump over here in red are these sage scrub species. So these are the species with scattered presence on the site that you would call sort of sage scrub-ish, or in some cases like Malacothamnus, it's really kind of a successional post-disturbance species, or Areodictyon is also a sort of post-disturbance species. So they cluster out pretty close to each other. And that tells us right away that what is present on these degraded sites is a pretty small subset of the trait spectrum that could be in this area. And then if we look at the sclerophylls, or the sort of, I'm going to abbreviate as HC, or hard chaparral species, we see that they cover a very big, much bigger trait spectrum. And that's consistent with what Steve Davis was saying yesterday about chaparral species really being very diverse. Okay, so if we filter just by drought traits, again, we see our sage scrub species sort of sticking close to each other, and our chaparral species spreading much farther out. I can tell you in the Q&A session if you want to know what traits contribute to the principal component axes. And I just want to point out here, there's an interesting little trend that Ceanothus oleganthus, which is one of two Ceanothi in the region, always clusters near Salvia apiana here. So it's kind of interesting that it's almost always clustering close to the sage scrub species, much more so than here's Ceanothus cuneatus over here, the other Ceanothus species. Can you guys see the arrows when I'm pointing? Yeah, okay. Okay, good. Okay, so then if we look at competitiveness, which is another set of traits in the restoration goals, and this is mostly traits related to plant size, but again we see and growth rates, the sage scrub species tending to cluster close together, Ceanothus oleganthus over near them, and the chaparral sclerophylls spread all over the place again. So if we put both drought traits and competitive traits together, again, we see the same thing. So some recurring patterns are that the salvias, we had 3 salvias in our trait database, Artemisia, Areodictyon, and Eriogonum are always clustering pretty close. So this is what's out there today, and it's a fairly narrow trait spectrum. The sclerophylls are very diverse. I didn't point out to you, but Sambucus and Encilia are consistently outliers on the fast growth end of the spectrum, so beyond where the salvias and artemisias and Areodictyons are. And that's kind of interesting. They have high specific leaf area, very thin leaves, low C to N. So you maybe could add them to this mix, but they would be potentially more successional. Ceanothus oleganthus is always clustering closer to the salvias than to the hard chaparral species. Maybe it's a good fit for the sites today. And then we see some species, which I'll just point out 2 of them, but there's a couple other pairs that always cluster really close to each other, Rhamnus salicifolia and Ceanothus cuneatus. And so when we're thinking about restoration, maybe we just pick the one that's actually easier to propagate instead of trying to propagate both of them. And that brings me to the final thing you do with this REST program, is that you have to filter the PCAs. So what we're proposing to do is to have 3 different community types. In the first one, we emphasize redundancy for these sage scrub type traits that appear to be successful today, but we add in some hard chaparral species that are closest to the sage scrub. So we're building a sort of redundant community. We're basically densifying and diversifying what's already there. And I put this in fancy writing so to encourage you to embrace the sage scrub vision. Okay, the second thing would be to maximize trait diversity across the entire trait landscape. So here we're maximizing the complementarity of traits rather than this idea of redundancy. So if you look across our principal components analysis, you pick things from all over the spectrum. And when you have pairs, like these 2 pairs right here, which is Ceanothus cuneatus and Prunus ilicifolia, or Rhamnus ilicifolia, you pick one of those 2 based on pragmatic needs and issues. So this idea is focused on maximizing the trait diversity across the landscape. And there's a lot of literature that supports the idea that highly trait diverse landscapes provide the greatest resilience to drought and potentially to fire, and maybe the best suppression of invaders. So this is really a hypothesis that we can test by contrasting these 2 different kinds of communities. And then the third sort of community would be what you might call a serial model, where we plant the sage scrub first, and then we add the chaparral species over time as later successional competitors. And there you might think that the sage scrub species could actually be potentially facilitators. And I'll just jump to what we're going to measure and say a little bit more about the possibility of sage scrub being facilitators. So we're going to plant these communities on a couple of these eroded grass-ridden slopes and measure survival over variable years. We're going to look at speed of canopy closure, biomass of natives, invasive grass cover, and colonization by non-restoration species. Some work that Stephanie Ma and Nate Moy have done shows that there is some native stuff in that seed bank, particularly some native herbaceous species. Now, another thing, so this getting to the issue of can sage scrub species be facilitators? We do have some places on the landscape today that are very dense patches of Malacothamnus fasciculatus. So one thing that Stephanie's doing as part of her dissertation is asking, can these existing sage scrub species act as facilitators? And so we're going into these stands. This is all Malacothamnus. There is exotic grass, in this case Bromus rubens, in the understory, and planting in a variety of hard chaparral or sclerophyll type species into either Malacothamnus intact, Malacothamnus thinned to 50% of the density, or completely removed, and then asking which traits are favored of these species that are getting planted in here under these different conditions. And this will tell us whether or not we can use these early successional sorts of species as facilitators in the landscape. So I'm going to conclude by saying we're taking this traits-based approach because we feel that it uses history as a guide but not a template. It gives us more flexibility on what to do. It provides a sort of quantitative basis for species selection, and it allows for hypothesis testing on the importance of traits. And it will create, hopefully, potentially multiple trajectories that on this different landscape. So maybe going back to our review here, we really are going to find out that what we have to do is embrace and enhance sage scrub. But we hope to be able to answer in 3 years from now whether or not, in fact, we can get a lot more hard chaparral sclerophylls on this landscape. Thank you.